Hi everyone, my name is Hania. I'm a research student at Sinai Health with the Coronavirus in the Urban Built Environment team, or CUBE for short. Today, we will be exploring various diagnostic tests. So let's say that a 65-year-old woman presents to the emergency department with fever, chills, a cough, and low blood pressure. Given her symptoms, you suspect she has an infection, and you order a nasal swab, a blood culture, and a rapid antigen test. So you've gone ahead and ordered all of these tests, but what are the differences between them and what are they really getting at? We'll be going over this in the next few slides. Nasal swabs and polymerase chain reaction. So nasal swab is so that you can capture the culprit organism and then it's analyzed using polymerase chain reaction. So PCR is used to make many copies of small sections of DNA or RNA. Each bacteria and virus has its own DNA or RNA signature, and by detecting the presence or absence of the genetic material, you can identify the cause of the infection. So how does PCR work exactly? Before a PCR assay can be done, DNA or RNA is extracted, and this is done to ensure genetic material availability. To amplify RNA using PCR, the RNA is first converted into complementary DNA using the enzyme reverse transcriptase. Then the PCR process follows these steps. 1. Denaturation. So heat is applied to separate the DNA into two single strands. 2. Annealing. So DNA primers attached to the single strands of DNA. 3. Extension. So Taq polymerase enzyme synthesizes two new strands of DNA using the DNA strands as templates. 4. Detection and quantification. So the amplified DNA amount is quantified, and the DNA strands can then be used to create two new copies, and so on and so on. So what are some pros and cons of PCR? Cons are that it's costly and it's susceptible to contamination and errors during the amplification process. Pros are that it's quick, simple, and highly specific. The second test that you ordered was a blood culture. To obtain a blood culture, a needle is inserted into the patient's vein, and approximately 20 milliliters of blood is collected and sent to the lab. In the lab, a sample of the blood is plated on a petri dish and placed in an incubator. The incubation period lasts up to 5 days, and if bacterial growth is observed on the dish, it indicates that the patient has a bloodstream infection. The exact bacteria is based on gram staining, which is a rapid dye technique, and further tests such as mass spectrometry can be used to identify the exact type of bacteria causing infection. Pros and cons of blood culture. Cons are that it's slow, so it can take days to see bacterial growth. Also, certain bacteria are difficult or even sometimes impossible to grow in the lab. Contamination from skin bacteria is also a possibility. Pros are that it's generally very accurate in identifying bacteria causing an infection. The third test that you ordered was a rapid antigen test. Rapid antigen tests can be used to detect viral or bacterial antigens responsible for an infection. How does it work? So a nose or throat sample is collected using a swab. The sample is mixed with a solution containing antibodies that bind to specific antigens if they are present. These antibodies are conjugated with a detectable label, such as colored particles. This mixture, now containing antigen-antibody complexes, is applied to a test strip with immobilized antibodies and chemicals designed to react with these complexes. The test line, denoted by a big T, shows a visible colored line if antigen-antibody complexes, labeled with a detectable marker, bind to the immobilized antibodies, and this indicates a positive result. Unbound complexes travel further along the strip to the control line, denoted by a big C, which shows a visible colored line confirming that the sample has migrated properly. Pros and cons of a rapid antigen test. Cons are that false negatives are a possibility, so it may not be able to detect antigens if a viral load is too low. Pros, rapid results, and it's very easy test to use. Other diagnostic tests. So multiplex tests simultaneously analyze a single sample for the presence of multiple targets, such as pathogens or genetic markers, using techniques like PCR, microarrays, or next-generation sequencing. One of the most common multiplex tests used in medicine are for RSV, SARS-CoV-2, and for flu. The second test is omics-based tests, which comprehensively measure the profiles of genes, proteins, or small molecule metabolites within cells or tissues. Here is an overview of the tests that we've gone over. So, nasal swab and polymerase chain reaction detects DNA or RNA of pathogens. It's highly specific and sensitive for viral and bacterial infections. It's also a quick and simple test to perform, but it's costly. Blood culture detects bacteria or fungi in the bloodstream. It's essential for diagnosing bloodstream infections, but results are slow, and some types of bacteria and fungi are difficult or impossible to grow in the lab. For a rapid antigen test, it detects viral or bacterial antigens. Results are rapid, which is great, and it's a very simple test to perform, but it's less sensitive than PCR, and false negatives are also a possibility. So after this presentation, I hope you have a better understanding of PCR, blood culture, and rapid antigen tests and can better cater to your patient.